Okay, so why should we care about the lac operon? Well, this lac operon was one of the first gene expression systems that was studied. And this was studied in the 1960s by three scientists, Jacob, Monod, and Waff. So these three scientists pictured here essentially worked out all of the details of this operon. And the most amazing part of this is that they were doing this without the modern biotechnology advances. So they figured out the different parts of the gene, the different elements that were acting, etc., without doing DNA sequencing, without actually analyzing the sequences. And the way that they did this was just through classical bacterial genetics. They worked out all of the details of this system. So the reason we still teach about the lac operon today is that this is an incredibly simple system that was the first to reveal some of the common features that we see in not only, for the most part, in prokaryotic gene regulation, but some of these elements we also see in eukaryotic gene regulation. These gentlemen coined the term operon, which, as we have discussed, is a group of genes that are under the control of the same set of regulatory sequences, and these are only found in bacteria. So let's take a closer look at what the lac operon is used for. So the lac operon is expressed only when a cell needs to use lactose. So normally, a bacterial cell prefers to use its preferred energy source, which is glucose. So the blank right here is glucose. In the absence of glucose, bacterial cells can use a number of alternative sugars, including lactose, maltose, arabinose, and they will use whichever of these sugars is actually available to them. So the bacteria will need to assess whether, first of all, whether glucose is present. If not, the bacteria will need to identify an alternative source for energy. The genes that are needed for lactose breakdown are only induced in the presence of lactose and in the absence of glucose. So if there's no ideal source of energy and if lactose is around, the cell will induce a set of genes that allows it to use lactose as an energy source. And this will include two genes in particular that we'll focus on, including a permease gene that allows lactose to be transported into the bacterial cell, as well as an enzyme that's called beta-galactosidase. So if we look at the activity of beta-galactosidase, it is the enzyme that will break down lactose into the two monosaccharides, galactose and glucose. So, only in the case where there's no glucose around will a cell need to turn on an operon like this to break down lactose. Okay. So let's look at the components of the lac operon system. The lac operon itself actually includes three genes. We typically focus mainly on the LAC-Z and the LAC-Y gene, but it turns out there's actually a third gene called LAC-A that are encoded in the lac operon. These three genes are considered to be the structural genes. These are the genes that get turned on all together through the control of one set of regulatory sequences which are found upstream of these three genes. So let's look a little bit closer at what these genes encode. LAC-Z, the one that we mostly talk about, encodes the enzyme beta-galactosidase. And as we've just looked on the previous slide, the function of that enzyme is to cleave lactose into galactose and glucose. The second gene that's part of the lac operon is LAC-Y, and this encodes the protein galactoside permease. This is that transporter protein that allows lactose to come into the bacterial cell to be used. Finally, LAC-A, which our textbook leaves out and doesn't really mention, encodes a transacetylase. And this For this course, we won't focus too much on this, but this enzyme puts a chemical modification on various lactose sugars. So these three genes are considered our structural genes. They will be turned on in the case that there's no glucose, but there is lactose in the cell. The other gene that is part of the lac operon, but is actually located upstream of the overall lac operon, is the lac-I gene. And this gene has got its own promoter, and so it gets turned on. This is actually a constitutive gene that is turned on all the time, and this encodes the repressor protein. That is one of the transacting elements that will come back and turn off expression of this lac operon if there's no lactose around. So we've got structural genes that are part of the lac operon. We also have the upstream regulatory sequences that are all parts that help to control the expression of the lac operon itself. So this is an overview of the lac operon that is taken from an image in your textbook, and this kind of simplifies to show us those two. Now it's showing 2 out of the 3 structural genes, including lac-Z, which is used to make galactosidase, and then lac-Y, which makes galactoside permease that allows the lactose to get into the cell. The other protein that we mentioned is the lac-I repressor protein, and as we showed in the previous schematic, lac-I is located upstream of the lac operon itself. This is going to make a protein, which is going to be a trans-acting element that will bind to one of the cis-acting elements upstream of the lac operon to control gene expression. So let's think for a moment about when a bacteria would want to turn this operon on. In other words, when would the cell want to transcribe and translate lac-Z and lac-Y? You might be thinking that the only time it would want to do this is if there was no glucose around, but there was lactose around. That would be when the bacteria would want to go to all of the trouble, waste all of the energy, or I guess not waste, but use the energy to produce lac-Z and lac-Y. When would the cell want to turn the operon off? Well, it would want to turn it off if it did have glucose around that could be used as the preferable energy source, or if there was no lactose. There's no point in making the enzymes to break down lactose if there's no lactose around. So let's take a closer look at how this works. What would happen in the absence of lactose? So we're going to consider a number of scenarios here and go through what would be happening at this lac operon. So in the absence of lactose, first question we have to ask ourselves is, would the cell want to turn this operon on? So no, it would not, because it would not want to waste the energy to produce the enzyme to break down lactose if there was no lactose there. So what would happen in the absence of lactose is that the lac-I repressor protein that is produced from the lac-I gene would act in trans, meaning that it would be produced and then it would bind to another element, and it would bind to a specific element called the operator site. So the operator, if you recall, is one of our cis-acting elements, and the operator is just upstream of the lac-Z and lac-Y genes. As you can see, when the repressor binds here, it physically blocks the RNA polymerase, which would have to bind at the promoter sequence, from crossing and transcribing the lac-Z and lac-Y gene. So in the absence of lactose, the repressor is bound to the operator sequence, which is right here, and this prevents the RNA polymerase from transcribing lac-Z and lac-Y. Let's take a closer look at the binding of the lac repressor to the operator sequence. This right here is showing you what the lac operator sequence looks like. And if you look very closely, you can see that the sequence at this side is exactly the same as the sequence on this side. So these two sites are called half sites, and it turns out that the repressor protein actually binds to the operator as a dimer. One of the subunits binds to this side, the other subunit binds to this side. So each molecule of the lac repressor binds to one half site. And this sequence is actually called an inverted repeat. Whenever you have the same sequence on this top strand as you do on the bottom strand, this is called an inverted repeat. So in the absence of lactose, the repressor protein is bound here to the operator. And how exactly does this repressor binding prevent transcription of the lac operon? Well, as the image in your previous figure showed, when the repressor is bound to the operator sequence here, this physically prevents the RNA polymerase from binding to the promoter sequence. So this image, this figure, which is taken from another textbook, shows the DNA sequence in the region upstream of the lac operon. So the +1 site is the beginning of the actual transcription site. You can see that the operator sequence overlaps with the +1 site. And you can imagine if you had a protein that was binding to this region right here, this would physically prevent the RNA polymerase from binding to the -35 and -10 sequence via its sigma factor. So repressor binding at the operator prevents RNA polymerase from binding at the promoter. The result of this is, of course, that there's no transcription of the lac operon genes. And this makes good sense because this is a time when the cell would not want to be making any of these LAC-Z or LAC-Y genes. This form of regulation is called the negative regulation of the lac operon. It's considered negative because the repressor binding is decreasing transcription of the operon. So this is a negative form of regulation which is mediated by the LAC-I repressor binding, preventing the transcription of the operon. So now let's consider a different scenario. So let's imagine now that the cell actually does have lactose present. We will consider whether or not glucose is present in a little bit, but for now let's just consider the case where we now have some lactose present in the cell. If lactose is present, lactose will actually physically bind to the repressor. So lactose is considered to be an inducer. The inducer or lactose molecule binds to the repressor protein, and this actually causes a conformational change, which prevents the repressor from binding to the operator sequence. So once it changes its conformation, you can see here it went from having stretched out binding sites here. It changes conformation once the inducer binds to it, and it comes off of the DNA. So at this point, there's nothing blocking the RNA polymerase from binding and beginning transcription of the LAC-Z and LAC-Y genes. Okay, so the RNA polymerase can then bind to the promoter and initiate transcription. So in what situation would you actually want to transcribe the lac operon? Well, we just said that there's kind of two things that the cell needs to be measuring. The first thing is whether or not there is lactose around. But the second thing that's even more crucially important is whether there's glucose. If there's glucose around, this cell does not want to turn on the lac operon because it prefers to use glucose. So how does the cell sense when glucose is around? We just looked at how it senses when lactose is around.